Yeah, welcome back to the channel, and today we focus on dihybrid crosses. Now the word di simply means two, right? So with dihybrid crosses, these are crosses that involves the inheritance of two traits at the same time. So the only difference between a monohybrid cross and a dihybrid cross is that in monohybrid crosses we deal with the inheritance of one trait. But with dihybrid crosses, we deal with the environment, with the inheritance of two traits at the same time, right? Now, let's try to focus on some examples involving dihybrid crosses. Now, in this example, it says that in the breeding season, male unknown lizards caught females by bobbing their heads. And it is said that bobbing their heads here simply means moving their heads up and down. That is what bobbing their heads means, right? While displaying a colorful throat patch. So they move their heads up and down, and the reason for moving their heads up and down is that at the same time they are showing a colorful patch on their throat. Now, it is said that the ability of anoles to bob their heads fast is represented by the allele capital letter B, meaning that bobbing their heads very fast is dominant, right? Over slow bobbing heads are small letter B, so they are recessive, right? So fast bobbing is capital letter B, slow bobbing is going to be small letter b. The allele for red throat patches, capital letter R, is dominant over yellow throat patches, small letter r. So, if we try to analyze this example, here we are dealing with the inheritance of two traits at the same time. Number one, it is the speed at which these unknown lizards bob their heads, and two, it is the color of the throat patch. So it is two traits that are being inherited at the same time, right? Fast bobbing, capital letter B, slow bobbing, small letter b, red throat, capital letter R, and a yellow throat patch is small letter r. Let's read further. Now, a male lizard, heterozygous for head bobbing. So if this male lizard is said to be heterozygous for head bobbing, it can only be capital letter B, small letter b. Now, again, since we're dealing with the genotype containing a capital letter and a small letter, the capital letter here will always win, right? So this male lizard here is going to have a fast bobbing. It will have fast bobbing because capital letter B is fast bobbing, small letter b is slow bobbing, and capital letter B is always going to win. And homozygous for red throat. And if it is homozygous for red throat, it means it is capital letter R, right? It is said that this male lizard mates with a female that is also heterozygous for head bobbing. So if this female is also heterozygous for head bobbing, it means that it is capital letter B, small letter b, with a yellow throat patch. Now, the yellow throat patch was said to be recessive, and in order for the recessive trait to be expressed in the phenotype, the genotype must be made of two recessive alleles, right? So if we try to combine everything together now, this male lizard is heterozygous for head bobbing, so it is capital letter B, small letter b, and it is homozygous for the red throat patch, which means that the genotype for the male lizard has to be capital letter B, small letter b. This is the heterozygous for head bobbing part, and this is the homozygous for the red throat patch part, right? So it is capital letter B, small letter b, capital letter R, capital letter R. They must be written at the same time. Now, it is very important that the alleles representing the same trait are written together, and these alleles are also written together. Now, the issue of having to write the genotype by writing the dominant allele first, hence capital letter B, capital letter R, small letter b, would be wrong in this case, because capital letter B and small letter b represent a different trait altogether. The same applies for these R's. So even though this is a small letter, they must be written close to each other like so, right? So it is capital letter B, small letter b, capital letter R, capital letter R. Let's look at the female lizard. It was said that it was also heterozygous for head bobbing, so it must be capital letter B, small letter b. This is heterozygous for head bobbing, and it was said that it had a yellow throat patch. So its genotype must be capital letter B, small letter R, small letter R. Now we must be able to perform a genetic cross with this information that was given, and let's try to perform this genetic cross. Now the structure of a genetic cross remains the same. It always starts with P1. In front of P1 now comes phenotype. It was said that the male had fast head bobbing. Fast head bobbing with red throat patches. That is the phenotype. And then the female also had fast head bobbing, but the difference here is that it had with a yellow throat patch, right? Below phenotype now comes the genotype. So the male was said to be heterozygous for head bobbing. So heterozygous for head bobbing is capital letter B, small letter b. And it was said it was homozygous for red throat patch. So it is going to be a capital letter R, crossed with. The female was also heterozygous for head bobbing, and it had a yellow throat, but the yellow throat trait was recessive, so it has to be small letter R. The recessive trait here is shown in the phenotype, and in order for the recessive trait to be shown in the phenotype, then the genotype must be made of two recessive alleles. Hence, yellow throat patch has to be small letter R, right? Let's continue. Below genotype now comes meiosis. After meiosis, now comes the formation of gametes. Now, this is where we have a bit of a challenge. With monohybrid crosses, we simply separate the genotype by a comma, but that is not practical here. In order to come up with the gametes for a dihybrid cross, you simply have to perform a cross involving the genotype of a single parent. What do I mean by this? So let's try to take this genotype here. It is capital letter B, small letter b, capital letter R, capital letter R. Now, if we separate this genotype, we will have a capital letter B, small letter b on one side, and we will have capital letter R on the other side. Now, how do we perform a genetic cross here? How do we come up with the gametes? This capital letter B will cross with the first capital letter R. Hence, the first gamete will be capital letter B, capital letter R, right? Let's come up with the second gamete. The second gamete, it will be the same capital letter B crossing with the second capital letter R. So the second gamete will also be capital letter B, capital letter R, right? Now, what about the third gamete? The third gamete will be the second B now. The second B is a small letter b. The small letter b will cross with the first capital letter R. So it will be small letter b, capital letter R. Lastly, it will be the very same small letter b with the second capital letter R. So it will be small letter b, small letter R, crossed with. We repeat the same thing here. So if we repeat the same thing, it will be the first B with the first R like so. So it will be capital letter B, small letter R. Again, it will be the first B and the second R. So it will give us capital letter B, small letter R. Again, now that we are done with the capital letter B, we come to the small letter b. The small letter b with the first R there. So it will give us small letter b, small letter R. Again, the very same small letter b with the second R there, and it will give us small letter b, small letter R. That is how we come up with gametes in a dihybrid cross. After gametes, now we come to fertilization. With fertilization, now we use a Punnett square. But since this is a dihybrid cross, it is 5 boxes sideways. 5 boxes sideways, and 5 boxes downwards. So 1, 2, 3, 4, 5. The top left box is always reserved for the word gametes. Now, since this is a dihybrid cross and we're showing the inheritance of two traits at the same time, the gametes must also have the two traits at the same time. Hence, we write it like this. So we're going to take the gametes of the first parent and write them at the top here. So it will be capital letter B, capital letter R, capital letter B, capital letter R, small letter b, capital letter R, small letter b, capital letter R. These are gametes belonging to this male parent. Now we take the gametes of the female parent and we write them below the word gametes. So it will be capital letter B, small letter R, capital letter B, small letter R, small letter B, small letter R, small letter B, small letter R. It will be like that. And then we come to this box. Let me change marking. So, and then we come to this box. To this box, we cross the capital letter B with the capital letter B. So it will be capital letter B, capital letter B. Now we come to the R's. We write capital letter R from the top and small letter r from the side. So it will be like that. Let's go down. So it will be capital letter B from the top, capital letter B from the side. So it will be capital letter B, capital letter B. Capital letter R from the top, small letter r from the side. So here it will be capital letter R, small letter r. I'm trying to write the genotypes that are identical with a similarly colored marker. Let me try and see if that will work. Now let's come to this box. In this box, it will be capital letter B from the top and small letter b from the side. So it will be capital letter B, small letter b, capital letter R, small letter r. The same will also apply to the very last box. So it will be capital letter B, small letter b, capital letter R, small letter r. Right. Let's continue. Now let's come to this line. When we come to this line, it will be capital letter B from the top, capital letter B from the side. So it will be capital letter B, capital letter B, capital letter R, small letter r. The same applies here. Capital letter B, capital letter B, capital letter R, small letter r. But when we come here, it is going to be capital letter B from the top, small letter b from the side, capital letter R, small letter r. So it will be identical to these ones. So it will be capital letter B, small letter b, capital letter R, small letter r, capital letter B, small letter b, capital letter R, small letter r. So that is what we have so far. Let's continue. In this box now, it is going to be capital letter B from the side and small letter b from the top, capital letter R from the top and small letter r from the genotypes for this box and this one has to be identical to this one. So let's write them. So it is the capital letter B from the side, small letter b from the top. So it will be capital letter B, small letter b, right? The capital letter R from the top and the small letter r from the side. So it will be capital letter R, small letter r. Here it will be the capital letter B from the side, small letter b from the top. So capital letter B, small letter b, capital letter R from the top, small letter r from the side. So it will look like that. And then we come to these boxes. So it is small letter b, small letter b. So small letter b, small letter b, capital letter R from the top, small letter r from the side. Now let's come to this one. Small letter b, small letter b, capital letter R, small letter r. So it will also look like this, right? Now let's come to this one. This one is also going to be heterozygous. So it is capital letter B from the side, small letter b from the top. So it is capital letter B, small letter b, capital letter R, small letter r. Here, capital letter B, small letter b, capital letter R, small letter r, right? Now let's come to this one. This one is going to be a small letter b from the top, small letter b from the side, capital letter R from the top, small letter r from the side. So this is in blue. So it will be small letter b, capital letter R, small letter r. Here, small letter b, capital letter R, small letter r. And that is all the genotypes that we have. Now, of course, below fertilization now comes F1. In front of F1, we simply write genotype, right? We simply write genotype. Now, let's look. How many genotypes do we have that are identical to this first one? So it is capital letter B, capital letter B, capital letter R, small letter r. It is these four that I've indicated in red. So there are 4 capital letter B, capital letter B, capital letter R, small letter r. There are 4, right? Now, let's come to this one that are heterozygous for both traits. It is 1, 2, 3, 4, 5, 6, 7, 8. It is the ones that are written in yellow. So we're going to write them as 8, capital letter B, small letter b, capital letter R, small letter r. The only genotype left is the one that is recessive for bobbing of the head and also heterozygous for the red throat patches. So it is these four. These genotypes are identical. So it is going to be 4, small letter b, capital letter R, small letter r. Now below genotype, of course, now comes phenotype. Now, let's look at these four. These four have got capital letter B, capital letter B. This is fast head bobbing. And because they've got capital letter R, so they are fast head bobbing with red throat patches. But what about these ones? These ones also have a capital letter B, so they are fast head bobbing. And they also have a capital letter R, meaning that they also have a red throat patch. So the ones that have got fast head bobbing, it is these 4 plus these 8. They also have the red throat patch. They also have the red throat patch. So even though the genotypes in this case is different, the phenotype is going to be the same. So the unknown lizard that have got fast head bobbing with red throat patches is this 4 plus this 8. So it is going to be 12. So we are going to write 12 fast head bobbing. 12 fast head bobbing. Fast head bobbing with red throat patches. Okay, so it is 12 fast head bobbing with red throat patches, right? It is this 12. What about the remaining 4? The remaining 4 have got small letter b, so this is slow head bobbing. But because they've got a capital letter R here, they've got red throat patches. So they are only different to these 12 because they've got slow head bobbing, right? So we're going to write 4 slow head bobbing. 4 slow head bobbing with red throat patches. Now, this is generally how we do dihybrid crosses. You simply follow the same structure and you will reach your answer very simply, right? Now, because we're already here, let's try to come up with calculation questions that may be asked, right? We may be asked, so since here we've got a grand total of 16 offspring that are produced, it is these 4, 8, 12, and 16. So the offspring that are produced from these two parents, it is going to be 16 offsprings, right? Now let's try to do some math. Let's say instead of the 16 offspring that are produced here, maybe there were, let's say, 32, right? Let's say there were 32 offspring produced. Then the question asks, if there are 32 offspring produced, how many would have fast head bobbing with red throat patches, right? So that is a very simple question to answer. Now, out of these 16 that we have, the ones that have got fast head bobbing with red throat patches, it is the 12 offspring, right? It is 12 out of how many offspring? Out of a grand total of 16 that were produced. Now, the question asks, if there is 32 that were produced, which means we simply multiply the whole thing by 32, right? So if we try to do our maths here, it is going to be 12 divided by 16, which will give us 3 over 4, and we multiply that by 32. And that will mean that the ones that will have fast head bobbing with red throat patches will be 24. Now, this is generally how we approach this question. Even if it said there were 64 instead of the 32, we simply say out of the original that we have, it is 12 over 16, and we multiply that the new number that they are suggesting, and we'll always get the right answer. That is how we do these calculations. Now, in terms of percentages, if they asked what is the percentages of offspring, for example, what is the percentages of offspring that are heterozygous for both traits? So if we are saying they are heterozygous for both traits, it is these 8. They are heterozygous for head bobbing. They are also heterozygous for the throat patch color. So it is going to be 8 over the 16. Because it is a percentage now, we multiply by 100. So this is 1 over 2 times 100. So our answer here will be 50%. Right? So that is how you are going to approach mathematical questions. You are going to approach calculations with regard to dihybrid crosses. Right? Let's continue. Now, these are typical examples that are usually set in the exam. Now, it says that coat color in mice is controlled by two alleles. Black is capital letter B, it is dominant, to gray, which is small letter B, okay. Tail length is controlled by two alleles. A long tail is capital letter D, a short capital letter T, and a short tail is small letter T. The Punnett square below shows a part of the cross between two mice. Genotype Roman figure number 1 has been left out. It is fine, we can find it, right? 1.6.1, it says give the genotype of parent 1. Now, this is a very simple question to answer. So we were given the gametes that are produced by parent 1 here, right? Now, this is an example of a dihybrid cross. We're dealing with the inheritance of two traits at the same time. It is the color of the fur, right? It is the color of the coat, and also the length of the tail. Hence, the gametes here have got two letters. The B here, it is for the coat color, and the T here is for the length of the tail, right? So, if this parent has got capital letter B here, capital letter B here, small letter B here, and small letter B here, it tells me that this parent for the color of the coat has to be heterozygous, because the gametes that are produced by the very same parent have got both the capital letter B and the small letter b. So in terms of B, it is going to be capital letter B, small letter b, right? Now let's look at the second trait. Now, in this first gamete, we've got a capital letter T. So it tells me that this parent's genotype also has a capital letter T. But in the second gamete, there is a small letter T, meaning that it also has a small letter T. And that is the genotype that is produced by the first parent, right? So we simply take the gametes, we'll look at the first letters, which is the B's, and we've got a capital letter and a small letter. So it is heterozygous. And then we look at the T's, we've got a capital letter and a small letter, so it is also heterozygous, right? Let's say maybe the question had asked for the genotype of parent number 2. We do the same thing. Now let's look at the B's. We only have capital letter B's here, which means that the genotype for parent 2 would have been capital letter B, capital letter B. Let's look at the T's. We only have small letter T's, which means that it is going to be small letter T, small letter T. This is going to be the genotype of parent number 2, and this is the genotype of parent number 1. Number B, it says give the phenotype of parent number 2. Now, the genotype of parent number 2, we've already come up with it. It is going to be capital letter B, capital letter B, simply because in these gametes here, there is no small letter B, meaning that it must be homozygous dominant for capital letter B, which is for black coat. And then we've got only small letter T's, meaning that it must also be homozygous for a short tail. So if we're to talk about the phenotype of parent number 2, it will be black coat color with a short tail, right? Black with a short tail. That is the phenotype, right? Number C, it says that give the genotype of offspring that is in Roman figure number 1. Now let's look at the offspring. Look at the offspring with Roman figure number 1, right? It is made from capital letter B on parent number 2 and small letter b from parent number 1. So we know that it is going to be capital letter B, small letter b, right? And capital letter T from parent number 1 and small letter t from parent number 2. So it will be capital letter T, small letter T. So offspring number 1 is heterozygous for both traits. Now we write this genotype like this because we know that when representing the genotype for the same trait, we start with the capital letter, then follow the small letter. So for the coat color, it was capital letter B, small letter b, and for the length of the tail, it was capital letter T, small letter t. It is not acceptable to write this genotype as capital letter B, capital letter T, small letter b, small letter t. It is not acceptable. The identical or similar letters must be written together as I've indicated here. Right. 1.6.2, it says, what percentage of the offspring above is gray with short tail? What percentage? Now, let's look at this. in order for them to be gray, they must be small letter b, because gray is the recessive trait, right? But neither of the offspring here have got gray tails, simply because of parent number 2, who only has the allele for black tail, which is dominant. So the percentage here is going to be 0%, because neither of the offspring have gray tails, have got gray coats. So the percentage is going to be 0%. 1.6.3. State the genotype of 2 gametes from the table above that will result in offspring that are heterozygous for both traits if fertilization occurs. So the genotype of 2 gametes that will result in offspring that are heterozygous. So we simply come to look at the genotype of the offsprings, right? Now, if we look at the genotype of these offspring from here to here, they are all heterozygous. They are all heterozygous for both traits. Now, the offspring in this row, they are made from this genotype of the gamete and the genotype from the side. So let's look at these individuals specifically, right? It is the gamete from the top, so it will be small letter b, capital letter T, and the gamete from the side, which is this one. So it is small letter b, capital letter T, and capital letter B, small letter t. We simply take them from the diagram. So this one was made from this one at the top, and also that one. So this genotype that is heterozygous for both traits, it is a combination of this gamete and that gamete. The same will also apply for any individuals in this line. It will be the same. So this is my answer. Next. It says that in a plant species, 2 characteristics, flower color and plant height, were studied. Each of these characteristics has 2 variations. Flowers may be red or white in color, and the plants may be tall or short. Plants that are heterozygous for flower color have got red flowers. Now, this is very important information, right? So if plants if these flowers are heterozygous, they will have red flowers. It tells me that the allele for red flower here is dominant over the allele for white flower, because in a heterozygous condition, the allele for red determines the phenotype and the allele for white is masked. Hence, I'm saying that the allele for red flower color here is going to be dominant, right? And the plants that are homozygous recessive for plant height are short. Homozygous recessive, it simply tells me that the alleles here that are used are small letters, and when we use the small letters, they are going to be short. So the allele for shortness is going to be recessive to the allele for tallness, right? It is going to be recessive to the allele for tallness. Let's look at the question. 3.3.1. What is the term given for a genetic cross involving 2 characteristics? We call this a dihybrid. We call this a dihybrid cross. That is a dihybrid cross. 3.3.2, they say, give the dominant phenotype for flower color. Again, we were told here that if this plant is heterozygous, it will have red flowers. Therefore, the dominant flower color is red. Red. The red flowers are dominant, right? Number B, the recessive phenotype for plant height. They already tell us that if it is homozygous recessive, it will be short. So shortness here is recessive, right? So the recessive phenotype for plant, it is going to be short. Number C, phenotype of a plant that is heterozygous for flower color and homozygous dominant for plant height. They already told us that if it is heterozygous for flower color, it will have a red flower. It will have a red flower. And if it is homozygous dominant, since when it was homozygous recessive, it was short. When it is homozygous dominant, it must be tall. A red flower, so it must have a red flower and be tall. So that is my phenotype. It will have a red flower and be tall. So that is my phenotype, right? D, genotype of a white flowering short plant. White flowering short plant. So we know that the allele for white flower color here is recessive, right? And we know that the allele for a short plant is also recessive, right? So this is a genotype that is homozygous recessive for both traits. And we know that from this table, they say that the allele for flower color that is recessive is represented by small letter f. So it will be 2 small letter f's. And we know that for plant height, the recessive allele is small letter h. And since it is said to be short, so the genotype will be small letter f, small letter f, small letter h, small letter h. That is the genotype. Right? 3.3.3, it says, state Mendel's law of dominance. This simply states that if an organism is heterozygous, then the dominant allele will determine the phenotype. The effects of the recessive allele are going to be masked. That is Mendel's law of dominance. In a heterozygous condition, the dominant allele determines the phenotype. The effects of the recessive allele are masked. Until next time, consider liking the channel or subscribing if you like what we have to say. Bye-bye.